I'm Cody Cabral here with Garrett Peltier, and today we're going to be talking about the effects of invasive shrubs on microclimate and tick populations. So it's known that plant species that are invasive can alter the characteristics of an ecosystem. In New England, some of these invasive species include privet and oriental bittersweet. These invasive species can grow so quickly and outcompete native species that they form dense invasive thickets, which can create their own microclimate conditions, resulting in ideal tick habitats. Ticks are a problem that exists in New England's forests because they are known to carry Lyme disease, which can be deadly to humans. So we want to know if the removal of these invasive species in these dense thickets will lower the number of ticks. The way we went about this is by first identifying a piece of land that would be suitable for experimentation on microclimate and tick populations. We selected the experimental forest behind Westfield State University because it has been the site of past experiments done by Westfield State students and is a reliable area for testing. The area was then divided into 3 different treatments where we would measure microclimate and tick populations. The native treatment is the control, and it's where there are no invasive species. The unmanaged area is where invasive species are the primary species in that ecosystem. The managed area is where invasive species had existed before but were removed as a part of a project by previous students at Westfield State. For each treatment, the microclimate was measured by hand by measuring the air temperature, soil humidity levels, air humidity, and as well as sunlight intensity. Then, within each plot, tick populations were also estimated by tick flagging method. This method involves a 2 by 2 foot piece of white cloth attached to a string that is run along the ground in a transect, which will pick up ticks as it's brushed along short shrubs and trees.
Hello, this is Garrett Peltier, and here's the results of our microclimate data and our tick collection. First graph above here, average microclimate characteristics within the treatments. We have humidity, air temperature in Celsius, and soil temperature in Fahrenheit to show the 2 differences. As you can see in the three different treatments, there is no real difference out of the average of the temperatures in each of the 3 microclimate collections we had. Below, you even see the majority of ticks were collected in the managed treatment compared to the other 2 treatments, unmanaged and native. Here is data that we collected from hobo loggers that we deployed in each of our plots. In the first graph above, the average temperature in Fahrenheit of all plots collected from the hobo temperature loggers, you can see there is once again no real significant difference in temperature in all of the plots. Where we begin to see a real difference from the data we collected is in the average light abundance or intensity collected in each plot by the hobo temperature loggers. One plot stands out significantly with a large variation compared to the other plots, with 886 compared to the closest one next being 475. After reviewing our data, we found that it does not support the hypothesis, so that therefore it is rejected. We also found that there is no significant difference in the microclimate measurements amongst the plots from all the data we collected and averaging them out. Concluded the only real difference we found amongst the plots was found from the hobo loggers in the light intensity collection. We also concluded that the highest tick abundance was actually found in the managed plots compared to the other plots in a very unexpected way. In further review of the results and data, we found that the results actually show a new possible theory amongst tick abundance in microclimate populations. We found that direct sunlight could be vital to tick abundance and its survival coming from the winter seasons into the spring. It could be for many reasons, such as the managed plots having no open canopy and an open floor where the direct sunlight can warm the soil during the winter months coming into the spring and speed up tick birth and tick egg hatching. I hope you enjoyed our project. Thank you very much for listening.